Okay, all right. Well, I'd like to welcome everyone on behalf of Laughing Whitefish Audubon to tonight's meeting. Our speaker tonight is Amy Munes, who conducted her graduate research on loons here in Marquette at NMU. Amy has a decade of experience in the field, ranging from work with seabirds on the northwestern Hawaiian island chain to conducting spotted owl research in the Pacific Northwest. Thanks for joining us, and I'll turn it over to Amy.
All right, thanks, Matt. It's a pleasure to be here. I'm glad you guys can make it. I'm a grad student at Northern Michigan University. I am going to be defending my thesis in probably about a month, and I will be get my master's. Alec Lindsay is my advisor, my research advisor, and this project was in partnership with Common Coast Research and Conservation. They did a lot of the work, like collecting samples for the wild loon populations. So I'm just going to give you a little guide of what the presentation is going to be. So I'll give you an introduction to loons. Not that they need an introduction, because I think most people know common loons. And then I'll kind of introduce telomeres a little bit, because I understand not everybody really understands telomeres that well. Genetic stuff can be kind of boring. I'm going to go over some of my methods, the sample collection, and the molecular methods that I went through, but that's going to be brief. I don't want to bore you with the details of that. And then I'm going to go over my chapter 1, which is telomeres over time. And that is going to just tell us like how telomeres degraded over the years. And I'll tell you more about that when we get there. And then chapter 2 is sibling competition and how that affects telomere lengths. Chapter 3, this is the big one, how methylmercury affects telomere lengths in the common loons. And then I'll go over some conclusions. So that's just a little guide to what you're about to see. So introduction to common loons. The Gavia immer is the scientific name. They are piscivorous water birds. So they forage in freshwater lakes, well, and ocean lakes when they're overwintering. But they dive, they eat fish. When they breed, they typically like oligotrophic, so low nutrient load lakes that are clear and crisp, so it's easier for them to see when they're foraging underwater. And those red eyes really help with their underwater foraging. They nest on islands and shorelines of North America. It's really good for them to have more vegetation around the lakes to help protect them from predators, which is important for their breeding success. And then they typically fledge 1 to 2 chicks per season. But that's starting to change as the ecology changes that they're breeding on. And that's part of why I'm looking at their telomeres. And another fun fact, they can live more than 30 years. So they're long-lived and they're pretty amazing birds. So throughout that lifetime, the 30 years, they can fledge up to 12 offspring, like successfully fledge 12 offspring in a lifetime. So their life expectancy is very important in having more fledglings and having more individuals, more recruits to the population. So why study loons? This is that Audubon Society map that I got online. There's a climate change report. That's where I got this from. You can check it out. It's a pretty cool graphic. But it shows their range in 2020. So the yellow is their breeding range, and the blue is their wintering range, and the green is the both, where they just stay year round. But as you can see in 2080, they're projected to be practically extirpated from the breeding ranges out of the United States. They're shifting north due to climate change and habitat loss and all that good stuff. So they're important. It's important to see like what's going on with these loons and I'm gonna look into their genetics. So everybody loves them. They're icons of the northern United States, and they might not be breeding here anymore, which is really sad, right? So another reason to study loons. So Walter Piper, I don't know if anyone has heard that name. He's done a lot of research on loons. He came out with a paper in 2020, and he found that the number of recruits have declined over the years. And not really sure exactly why. Like, we saw that it's happening, but there are a lot of reasons why, likely. They may have moved further north, as you saw in that map from before. Like, they may have just been shifting north. So maybe the recruits are going to more inhabitable lakes for breeding that they know will be more successful. Yeah, so habitat availability is a big problem. Eutrophication, so like nutrient load, is a problem. I'll get more into that in the next slide, further down. And then another problem is their fledgling rates have declined. And this is also a graph from a Piper paper. So the fledglings over the years have declined. It's only been from 1993 to 2019. So this isn't, I mean, it's a long-term study, but in the grand scheme of things, we don't know what their populations have done before 1993 or like over the years. So this could be like, these could be natural fluctuations, but the trend is more towards they're just moving out of these areas, more people are moving in, there's more shoreline development. So they're seeing, there was a 26% decline in their fledgling success rate, even though their nesting attempts did not change. So they nested the same amount, but fledged less chicks, which is kind of, like, what's going on? Also, the mass of the chicks has been declining. So that tells me that there's probably less food availability for them. So I'll get more into why I think that is in the next slide. So some of the potential causes that I think is going on is habitat loss, shoreline development is encroaching on their habitat. Like I said, they need a lot of vegetation around their shorelines to be able to have protected like nesting sites. So that is causing like less habitat availability. People like mowed lawns down to the lake. That's bad for loons. People like outdoor recreation, so they'll be kayaking out on the loon. Oh yeah, I love that majestic loon, but let's boat up close to them and see what they're like. And that affects them, and they're less likely to come back and breed on that lake if they get disturbed repeatedly. So outdoor recreation, as much as I love it myself, it can just be disturbing to the loons. Hey, Alec. And another potential cause that I believe is happening is like eutrophication, so increased nutrient load in the lakes, making it more turbid. So the loons are having a harder time finding prey, like the small prey that they can feed their chicks. So they like to have clear lakes because it's easier for them to see. They need to be able to see what they're getting for their chicks to eat. The smaller fish are harder to see, then they end up feeding them nutrient poor, like invertebrates and stuff. So that might be contributing to the decline in their mass over the years. And just like the lack of fledgling success. Yeah. Oh yeah, and the lower pH is also a problem that I think is happening. So the lakes are getting more acidic, and that's leading to higher methylization of mercury in the lakes. And the methylmercury can move up the trophic levels more easily than mercury itself. Sorry, I'm getting a little shaky. I'm very passionate about these loons. So methylmercury is also a problem that they're seeing in loons. It increases the stress of the loons and could potentially be causing more decline. So it's not just one thing, it's a multitude of problems that is happening to these loons. And I think a lot of these things are going to be reflected in their telomere lengths of the blood. So that's what I looked at for my master's research. So what is a telomere? It is on the ends of the chromosomes. It's the lit up portions of this little photo here. It's a non-coding portion of the chromosome that kind of protects the important coding portions from degradation, because through each round of mitosis, a little bit of the genome is lost, or a little bit of the telomere is lost, and it's just the natural process of mitosis. So with every round of cell division, a little bit is shaved off. This is a little bit exaggerated, but you get the idea. So over time, we all lose, we all have telomeres. Over time, they just get shorter naturally. But in addition to the natural causes of telomere length degradation, stress. Stress can also cause the more rapid degradation of the telomeres. And once the telomere is degraded to a certain point, the organ starts to fail. And then you see the cells senesce, the organ fails, and then the organism ends up dying, unfortunately. But the theory is that ROS, reactive oxidative stress, the reactive oxygen species, comes in and it disrupts the replication of the telomeres. And this is theoretical. I don't think it's anyone really knows for sure what is going on, but so the ROS comes in and disrupts the replication of the telomere, and it kind of loops, it creates this loop. And as a defense, it slices off that section of the telomere, so it increases the rate of the degradation of the telomere, and stress causes this. So it increases, that's what is increasing the rate of degradation. So they say. So for my thesis, I looked at how age impacts the common loons. I looked at how the sibling status affects common loons, the larger versus the smaller, because the smaller are going to have less of the feedings probably, because the larger is going to be more aggressive. It's going to be able to get to the adult when the feeding comes. And if there are less feedings, that larger individual is going to get more of the feedings. The smaller individual is going to get less and have more stress and lead to more telomere degradation was my hypothesis. And then how mercury affects the telomeres through stress. So the sample methods, a lot of the samples were collected by Common Coast Conservation, Research and Conservation. They collected samples from all over the UP. Most of them are from Seney National Wildlife Refuge, but a lot of them came from other places. About 50 samples came from various places throughout the UP. And I'll show you a map in a second. I had additional samples from a study that was done by USGS. Kevin Keno did a mercury dosing study where he hand reared some loons, fed them mercury, different doses of mercury, and took blood samples after 15 weeks. So I used those blood samples to see how the mercury affected, in a controlled study, affected the telomeres. And I have to say that the Museum of Zoology had all of these all these blood samples to do this telomere study, and they have a very impressive collection. And I just have to thank the museum curator, Alec, for allowing me to use these samples. It's an amazing collection, and I was very impressed. And that's largely what brought me here to do this research. Here are the sample sites that CCRC, where he collected his samples from. The red dots show like the higher mercury levels in the loons, and the yellow dots show like the lower mercury levels. So it's the mean mercury level from the loons that were collected at those sites. So it just kind of shows which ones have the higher mercury. So here's Sini. Yeah. So Sini is, I can't show you the one on Zoom, but I guess I could. There's Sini. It's a very protected system. So it has like lower, it has more of a neutral pH, and the methylmercury is not really a problem, but a lot of other lakes throughout the UP have pretty high mercury loads, or at least the loons had high mercury loads. So for my methods, I extracted the DNA from the blood, used quantitative polymerase chain reaction, amplified the telomeres using telomere primers, amplified a really conserved single copy gene, and calculated the telomere single copy gene ratio to kind of get the telomere length. But I'm not going to go into like the great detail of how that happened because it's kind of boring. And yeah, that'll be for my defense. If you want to know more, come to that. So now I'll get into chapter 1 results. How telomere shortened over time. Actually, I should go back. So they naturally shorten through mitosis. And this helps to validate my assay to make sure, because this is molecular, right? So I was like, I needed to do something to make sure, okay, this seems to be working. So I looked at 9 individuals that were sampled twice throughout their lives. And they were sampled several years apart. So 6 were sampled as juveniles first, and then later as adults, years later. And then 3 of them were sampled, but like as adults first, and then again later as adults. So here are some of my results. So these are each individual is right here. So you can see the first measurement and the second measurement. And all but one declined, which is a good indication that my assay worked. There's the one that didn't decline, but I'll talk a little bit more about that in the next slide. But you can see overall, the telomere lengths got shorter over time, which is a good sign that this worked, like I said. Significant results right there. So yay for low p-values. But interestingly, it looked like the initial telomere length affected the amount of telomere degradation we saw in the loons. Yeah, so if they had longer telomeres, they had more degradation. If they had shorter telomeres, they had less degradation. And with that individual, this guy, the one that went up, there are a few reasons why that could have been. It could have been like something went wrong with the assay, but I mean, it's possible. I mean, it's very possible that happened, but it could be something else. So in penguins, they found that right before the penguins fledge, the telomere lengths increased. So maybe when the sample was taken, they got it, like maybe loons see like a similar thing, like maybe their telomeres increase like right before they fledge, and we just kind of caught it before that increase happened. It's hard to really know what happened there, but as you can see, like in that trend, the initial telomere length affected it, and you can see the initial telomere length is really low, right? And look, it fits really well on that trend line. So that tells me that I'm not sure if it was the assay or if there is something going on there. So that's something that could be like further researched, like to see like at different times that the loons, you know, at different times in their juvenile life have does the telomere change over the growth period? Yeah. So I found that interesting. But it's pretty strong correlation right there. So chapter 2, I looked at how like sibling competition may be affecting the telomere lengths of the individuals. And my hypothesis is that larger siblings will have longer telomere lengths because they're getting more feedings, they have less stress, the smaller juveniles are getting picked on by their older siblings, and this is causing more stress. Like I said, the oxidative stress sloughing off a lot of the telomeres more rapidly than they should be. And yeah, so just the stress from being little could be increasing the rate of degradation. So I didn't see much of a difference between the large, so the L is the large, the S is the small. Not a very big difference between the two. That's, I mean, if anything, there's like a slight longer telomere in the smaller individuals. But when you look a little closer, there is a trend that is interesting. So this is the mass on the bottom, and then the telomere length on the axis, and you can see the large and the small, LS. And as the mass increased in the small individuals, their telomere lengths got longer. So the smallest of the small have the shorter telomeres, and those are going to be the most stressed out birds, right? So this kind of supports my hypothesis that the small individuals have higher stress and their telomere lengths are degrading faster, because you don't see that in the large individuals. As the large individuals get larger, their telomere lengths get shorter, which seems, so with rapid growth, a lot of juveniles, when they're rapidly growing, they have high rates of telomere degradation because they have high rates of like high cell turnover because they're growing, right? So their telomere lengths will start to decline as they get larger. So I'm thinking these larger individuals went through that more rapid growth phase. So their telomere lengths are getting like slightly shorter. So they're on a downward trend. Then these small individuals, if there wasn't some sort of stress implication in my mind, they should show the same trend, right, as the large, but they're showing the opposite. So it's not a super strong, you know, my R square value, it's not super tight, but there's definitely an obvious trend in that direction. So there is some support for that hypothesis. And then I looked, so anecdotally, I kind of looked at some of the individuals across year siblings. So that's just two breeding pairs that have had several offspring throughout several years. And I looked at the telomere lengths of each offspring over the years. And as those adults got more experience with their breeding, it seemed like the telomere lengths got longer. So that is an interesting trend. There are not very many data points. So this would need to be researched a lot more. This is more anecdotal than anything because I had the data. I don't have much data, so I don't feel super confident saying like, that's a fact, because more research would need to be done. But it looks like a pretty strong correlation there, right? So yeah, more experience. Loon pairs have higher quality offspring, according to this anecdotal analysis that I did. So on to chapter 3. This is what everyone's been waiting for. I don't know if you guys, the methylmercury, like how the methylmercury affects telomeres. And I believe that the methylmercury is going to decrease the telomere lengths because past research has shown that methylmercury increases the stress load of the loons. Therefore, I believe that it would decrease the telomere lengths. Interestingly, oh yeah, I need to go into the mercury. I had to put this up because I made this and I liked it and I wanted to show you guys. So it's just the mercury cycle. So mercury is expelled, is bioaccumulated in the system up the trophic level. It is introduced to the environment through industrial processes in high volumes. Like it's naturally occurring, but it occurs in higher volumes because of these industrial processes. And then it sinks to the bottom because it's a heavy metal. These little microbes convert it into methylmercury. So mercury itself does not accumulate up the system, but once it's methylated, it bioaccumulates. So these little sulfur-reducing bacteria and archaea convert the mercury to methylmercury. And they're more prolific in lower pH lakes. So more acidic lakes tend to have more of these bacteria. And then, so like as the lakes acidify, we're seeing like more and more of this as a problem. So then they get eaten up by little inverts. The inverts get eaten by the fish get eaten by larger fish, and then the fish, you see how that works. And as it bioaccumulates up, loons are at the top of the trophic level in these systems, in these aquatic systems. So they have the higher end of this mercury load. So they're a good study species for mercury effects. And then I lysed the blood after CCRC collected these samples. I lysed the blood, extracted DNA, ran qPCR, and measured those telomeres. So for the laboratory study, these are the different dosing regimes that Kino used, like the USGS study that I talked about. So they fed them 3 different doses, and then there was a control. So there's a 0.08, 0.4, and 1.2 micrograms per gram wet weight of the loon. To see, but you can see that it didn't seem to affect the telomere lengths here. There was more of a spread, which is interesting. So like the more dosing they got, the more of this, like the more varied the telomeres got, which is kind of weird. And then the control looks pretty, like the control and the 0.08, the lowest dose, they're very similar in the spread and in this data as well. So the bottom is the blood mercury levels, because they were sampled at 15 weeks, and then they tested the blood mercury of each different dose, and then I measured the telomeres of each individual. And you can see that, and these are the doses. You can see the control and the 0.08 dosing regime treatment. Didn't really see much variation with the telomere lengths, or it's very clustered. But you can see in the control, there seems to be a little bit more spread, or not the control. In the 0.4, there's a significant slope showing like an increase in telomere length. Yeah, I don't know. I don't know. That was unexpected. I was expecting it to go down, not up. So I'm not really sure what that's about. And I believe in the 1.2, we would see the same trend if I had more samples. I only had 8 samples, so you could see there's starting to be a trend in that same direction, but I didn't have very many samples. I wish I had more samples altogether to like have more robust results, but it's an interesting start. If somebody else were to look further into this, this could help guide them in the right direction, I guess. So yeah, that's the most interesting graph of the graphs in my mind. And that's significant, low R or low p-value. So yeah, I don't know. I wish I knew why they would get longer, but it's more, I haven't seen much literature about how mercury affects telomeres. It's kind of hard to find. So moving on, in the wild populations, this is all of the samples that I tested. And the colors are the pH. So the higher the pH, or the higher the pH, the more neutral. Those are the lighter colors. The darker colors have the lower pH, so they're more acidic lakes. And I found this interesting. You can really see the blood mercury correlation with the pH levels. So as the pH gets more acidic, the loons have more blood mercury, but it doesn't seem to affect their telomeres in a significant way. Yeah, I mean, I thought I'd find something more interesting, but this is what I found. Doesn't seem to be, even when I break it down, I don't find any significant trends when I look at the non-refuge individuals and the CNE National Wildlife Refuge individuals. So these are the non-refuge and these are the refuge. And it doesn't seem to show any trends. But that doesn't mean that it isn't affecting their telomeres in any way. If you looked at different tissues, such as the brain, because mercury bioaccumulates in the brain, and that is what affects them the most, because it causes them to be less attentive to their offspring. They just are, it just causes nesting failures largely. So I think if you looked at brain tissue, you might see something different, but in their blood, it doesn't affect their blood telomeres, but mercury is only transported through the blood. It doesn't affect their blood. So I think if you looked at different tissues, you might see different results. And in my conclusions, telomeres get shorter with age, pretty definitive. Initial telomere lengths influence the rate of degradation. That seems to be pretty solid. Smaller loon chicks have shorter telomeres. In the smallest of the small, seems to be the case. There's a trend in that direction. Parental experience leads to chicks with longer telomeres. More research needs to be done. Not quite sure if that's a thing, but it looks like it is. Preliminary research kind of shows that. And methylmercury shortens telomere lengths in blood. No, it doesn't seem to. So with that, I'd like to thank Alec Lindsay, Kate Teeter, which couldn't be here today, unfortunately, for giving me guidance on this project, and NMU for helping providing funding. I want to thank Common Coast Research and Conservation, who is like, this wouldn't have been possible without their data collection and blood collections. USGS for letting us use their blood samples from their mercury dosing study, and Zach for listening to me give this talk several times and putting up with me. All right. And with that, I'll leave it with questions. Thank you.
If you look at populations in Canada, which perhaps are more pristine, do you see the same patterns that you're discussing here?
I can't say. I mean, we would have to look at those individuals, but I think it would be worth looking at different lake qualities, like getting the turbidity of the lake and like taking lake measurements. I didn't really get in the field and do a lot like any of that research, but I feel like this is a good start, maybe open the door for somebody to kind of get into more like lake quality implications with these telomere lengths, and maybe get into Canada and look at those lakes, like look at the really super pristine lakes that aren't disturbed by humans, look at all different levels. And that's the only way to know how like that is gonna, like these are all just like, I think these are the causes of the telomere length differences and the causes of like the declines, but like nobody really knows for certain. Yeah.
So just to say this related, to that, so as far as the genetic component, we don't know, I think it's the first one to look to.
Oh, yeah.
But and the mercury piece is definitely in Canada, so even though, so if you look across, west to east in Canada, the mercury loads in loons increase as you go closer to the Maritimes, the Eastern Maritimes. And it is, so there's both a pH piece there, like, so, right, low pH lakes, there's a lot more of those sulfur reducing methanogenic bacteria that will fix the mercury to make it methylated and then enter the food chain. But it also is almost certainly due to the deposition by predominantly coal-fired power plants.
Yeah.
That all those effluents are making their way predominantly from west to east.
Hmm.
And so as you go, even as you get up in Nova Scotia and Newfoundland, we find some of the blood mercury in those loons, even though they're really pristine lakes, but you go in and sample their bloods and they're the highest in North America. And they then have lowest productivity.
Ah.
It's actually one of the things that's motivated some of this research for a long period of time is that people did a lot of behavioral analysis. In fact, this is actually where I got my start with loons over 25 years ago, was doing behavioral studies of loons that were on high mercury lakes and low mercury lakes. And we can't tell the difference, like parents that have high mercury levels and those that have low mercury levels, they weren't doing these, according to time activity budgets, all this analysis we did, we couldn't see anything different, yet they were fledging fewer chicks. There was something going on. And so that's part of the reason this, like, Kevin Kenow did that study in 2003 and 2007, and it's going all the way up here to Amy's work.
The sampling of the mercury in those various provinces, I would suspect they would say that, and you were using red blood cells as your sample base for the telomere.
Hmm.
I wonder if they would still have that would be accessible.
They might. Blood mercury is sometimes the case, but they also use a very specific feather. So
Hmm.
the secondary, they will take that. One of the interesting things about loons is that they shunt. So when they have that circulating mercury in their blood, during molt, they will shunt that mercury into their feathers. So they'll actually have really elevated levels of mercury in their feathers. So it's a way that they actually get rid of mercury out of their system and stick it in your feather. It's like a waste product that they can put it in and get rid of it. So those feathers they may have, and so a lot of them are sampled with feathers and not with blood.
Yeah, I do have some.
Yeah.
That'd be interesting.
It would.
Yeah, I do have some feather mercury, but not a whole lot. Like, yeah. Mhm.
How do we know that it's the initial lakes that we're taking samples from, if they're migrating or changing? Like if there's any outward influence, if they're going to different lakes, are loons pretty solitary in their area year round?
No, they'll like breed, they'll kind of stay in the same area during breeding season because they don't want to get too far away from their chicks. But then they do migrate down, and I meant to mention that. I guess I forgot to say it. They migrate down to like the Gulf of Mexico and the East Coast United States.
Okay, so are you just trying to pick up where where this mercury is being accumulated from in the north?
So a lot of these individuals were juveniles and they've only lived on the lakes that they were raised. They haven't migrated yet. So a lot of these were very isolated to the lakes that they were raised on. And the adults will go to other lakes to forage and feed their chicks, but not very often, and only if there's very low foraging ability in their breeding lakes. So typically they forage pretty close to their juvies. They don't want to leave them too far.
To follow up on that, so if you look at some of the original papers that were looking at the mercury levels of adults and chicks, and you compare the mercury level of a pair of adults on a lake in their feathers and in their blood mercury, and you look at the chicks on the lake, like Amy was saying, so the adults can have a lot more variable blood and feather mercury, in part because they've been living for 20 years. There are years that just spent the chicks are like, it's a spot on.
Mhm.
So it's very spot on for the mercury in the lake, actually it's the pH of the lake.
Mhm.
The chicks will have a really tight correlation. to
Yeah.
mercury.
As you move, oh, please.
No, yeah. Hannah?
What is the primary driver of the acidification of the lakes?
Well, I think, like, so a lot of, like, the acid rain, I think is a lot of this, the pollution that's coming from these power plants, they're like putting a lot of carbon into the atmosphere, and it's like causing a lot of like acid rain, they call it. Well, I mean, it is, and it's causing more acidic lakes, largely because of the runoff. I believe that's like the large cause of, I mean, it's a, like,
Not so much.
growing up.
in the UP.
yeah,
It's very
okay,
true out east. It's
Okay.
much more true out east, but in the UP and a lot of our lake, I mean, there's just a lot of different geomorphological, geohydrological conditions. And so like, if you go on a high boggy lake, a bog lake is gonna be relatively acidic.
Oh, yeah,
Very
yeah.
low pH, and it just has to do with the geomorphology of it. So
okay.
whereas like down by Escanaba,
Oh,
or
yeah.
something where you have a lot of limestone,
Mhm.
those lakes, the limestone base is gonna buffer that a lot better.
Yeah.
So it's a lot of times on these kettle lakes that you get in northern Wisconsin and in the UP. So they don't have a lot of inputs and outputs, water's not flowing. Flowages are usually a lot better shape. But if you have a kettle lake where it's really well contained, spring fed, a lot of granite, it ends up being really low pH.
Yeah,
And
yeah. Thanks, Alec. All right, anybody else? Any Zoomers?
there's a in the chat.
I don't, yeah, I don't. Yeah, I only have my dad and my friend Amy. Nice to- Hey, Dad. But yeah, all right, we're good.
Thank you.
Thank you, everybody. Thanks. All right.
Your meeting is done.
I think
Interrupted.
so.
That's it.